you really have been, while leading your own, great lab, you've been a very collaborative scientist. Within Duke, you and I have worked together on cardiovascular disease together, but also outside of Duke. Can you talk a little bit about how you view team science and collaborations?
Yeah, absolutely. I think one of the main things that I'm sort of known for, and we can get on to talk about single cell and spatial, and people just assume that I work on just direct the core, but my own science has been heavily involved in multiple sclerosis and then Alzheimer's disease, autism, and then brain tumors more recently. And it's pretty apparent that we can't carry out this sort of science as basic scientists from my own perspective without the engagement with physician scientists, without clinicians, without statisticians, bioinformaticians, etc. And so for my own neurological disease research, those collaborations have just been fantastic for pushing the science forward. And again, it's sort of this team science approach. With respect to the core, I don't see the point in developing or instituting technologies that don't benefit everyone. There's no point in being siloed away with respect to technology. I think it's, the rising tide sort of analogy benefits all those who are carrying out disparately related science, but the technology may be really important for other people you just haven't thought about.
Yeah, Simon, and I think what you bring to the table, right, as a molecular geneticist, a basic scientist, but also somebody who understands, even if you're not a clinician, you understand the clinical questions that need to be asked. For example, in your multiple sclerosis work, I think it's really fantastic that you have this diverse expertise, but then you pull in teams so that you can really exponentiate science. And I think it's a perfect model of how we need to think about genetics, genomics moving forward so that we have the largest impact on human health. So just kudos to you for being able to do that. On that note, can you talk a little bit about your core? You're really one of the world's experts on single cell, single nuclei technologies. Can you talk a little bit about the work that you do in the core and how you collaborate in that space?
Yeah, sure. So I've always been a bit of a tech junkie. So when I was at the Sanger, myself and a colleague who's now at the Jackson Labs developed a method of fluorescently mapping or constructing physical maps of genomes. It had been done radioactively until that point. So we transitioned that radioactive technique into a higher throughput fluorescent technique. So that was used on a fairly large proportion of the human genome. And I think ever since then, I was interested in sort of novel technologies and how they can be applied more broadly to science. I think if I think it's a good idea for my own neurological disease research, I feel like other people might be interested in it. And so it's a matter of seeing what technologies are really transformative. Identify where there's an opportunity with these novel technologies, how we can change the paradigm from approaches that we've been using traditionally for a long time, apply these technologies that give us a unique aspect on the science and be able to understand the mechanisms of science. And I think that's why, we were one of the first labs on the East Coast, I think, to get into single cell with 10x Genomics. We also maintain that relationship with 10x by adopting spatial transcriptomics approaches. So not just looking at very high resolution single cell, but putting it in a spatial context. And then we were the fifth group in the world to have 10x's Xenium platform, which is their in situ sequencing platform. And then we most recently were the first independent use of the platform in understanding diffuse midline glioma. So it's sort of this transition of getting from where we used to be doing particular approaches to understand science to really adopt these novel technologies to ask novel questions.